So tonight's video is going to focus on enzymes. This should essentially be a complete review from your freshman year. There's not too much new stuff here. Again, though, the goal is going to be to take this material and apply it in different situations and experiments, not just spit back a couple of facts. So first we're going to look at some properties of enzymes, both structural and functional. So a couple of things that I want to remind you guys is of the fact that enzymes are biological catalysts. Remember, a catalyst is something that is going to speed up a chemical reaction. So they will speed up reactions, and remember, they're going to do that by decreasing the activation energy. We're going to look closer at that later. So they will lower the activation energy, which will in turn allow the chemical reaction to speed up. Remember, they are reusable. So an enzyme is not consumed every time that it is used. It can bind to another substrate, and then another one, and then another one, and another one. So they are reusable, and remember, they are proteins, right? And remember some of our structures of some of our components of proteins, right? Proteins are made of amino acids, so they're polymers. They're large molecules. This large blue right here, this is our enzyme. So it's a large protein molecule. And remember, its shape is going to be very important. Remember, when we were looking at protein shape, that was an issue. The primary structure, the order of those amino acids, that secondary folding, either into that beta-plated sheet or that alpha helix, followed by that final tertiary folding based on some of the properties. And when that protein loses that shape, it doesn't work properly anymore. And so that will come into play with our enzymes as well. So they're reusable proteins that are going to speed up our reactions by lowering the activation energy. If y'all remember from freshman year, they have an active site. You can see the active site on this enzyme right here. The active site is where the substrate is going to bind. So our substrate in this example here is this little red molecule here. So the substrate is what will bind to the enzyme, and the active site is where it will bind to the enzyme. And remember, our enzymes are substrate specific. So they're looking for a particular substrate that fits into their active site. They can't just work with any substrate that they want to work with. So when our enzyme finds a particular substrate that it is compatible with, these enzyme-mediated chemical reactions can occur. So these particular chemical reactions, they're just saying that these are the reactions that are occurring when the enzyme and substrate are able to bind with one another. When the enzyme and substrate do join together, it's called the enzyme-substrate complex. And the enzyme will adjust its shape a little bit to make sure that it gets a nice, perfect fit. You'll notice here how it kind of closed around. And when that happens, that is what's called our induced fit. So we induced a perfect fit. We caused a perfect fit. So that induced fit will happen as that substrate enters the active site. The enzyme will change shape just a little bit because of the interactions between the side groups that are on the amino acids that are in the enzyme, as well as the chemical properties of the substrate. That will cause that enzyme to shift its shape a little bit, resulting in that good, tighter fit for that substrate in that active site. So enzymes are going to be used to help regulate biological processes. Their structure and their function are what enable them to do this. You can see up here on the top, we've got our substrate, and here's our enzyme substrate complex. Our enzyme here is sucrase, which is working with the sucrose substrate here. So the sucrose is able to bind with the enzyme here at the active site, and you get the induced fit for the really nice tight fit there. The substrate is then broken down into two molecules, okay? And then you see the enzyme getting ready to be used again. So they're reusable. They made this reaction of breaking the sucrose down into individual glucose molecules happen a little bit faster, and they did that by lowering the activation energy. Hopefully you remember this graph from before, right? So the black line here is the energy that was required to get the chemical reaction happening without the enzyme, where the red one is the energy required to get the chemical reaction happening with the enzyme. Remember, our activation energy is basically it's our energy needed to start our chemical reaction. And so if that energy is higher, then it's going to take our chemical reaction a little bit longer to occur because you had to work harder to get it started. But if we can lower that activation energy like you're doing with the red line here, then that reaction can happen a little bit faster because you don't need to work as hard to get the reaction started. So that activation energy will be lower in reactions that are mediated by enzymes. But what happens if something goes wrong? So if the enzyme structure changes, just like when we talked about a protein changing, because an enzyme is a protein, if that enzyme loses its structure, then it can no longer do its job. And that specifically is called a denaturation. So when that enzyme denatures, it gets a change in the shape of the active site. So the active site shape is no longer the same as it was before. Therefore, the substrate would not be able to fit into it. And so when the enzyme denatures, it can no longer help or catalyze chemical reactions. So when it gets this far, the temperature is really high, the pH is off, the enzyme then can no longer catalyze the chemical reactions. It doesn't help speed them up because the substrate isn't able to bind. And so that's another example, like we've been mentioning as we've been going through, is this structure-function relationship. The loss of the enzyme structure results in the loss of the enzyme function. So because of that possibility of the enzyme denaturing, enzymes are going to work in an optimal range, an optimal environmental range. And that optimal range is going to be whatever point at which you can have the maximum amount of interactions happening between the enzyme and the substrate. Okay? And so there'll be a window for certain enzymes. Like enzymes in our stomach will work better at a low pH, but enzymes in our mouth don't work really well at that low pH. So there will be optimal ranges, optimum environments for different enzymes. But what that means is that they are at their most efficient. They are binding to as many substrates as they can, as quickly as they can. That's their optimal range. So when it gets out of that range, right, when the temperature gets really high or the pH becomes really acidic or really basic, then it becomes outside of that optimal range and the enzyme can denature. It can lose its shape and lose its function. So let's back up a second and talk about temperature specifically, and then we'll look at enzyme. So if we're looking at temperature, and you're looking at this graph here on the left, okay, the black is the optimal temperature for a typical human enzyme. Okay, this is in Celsius, so I know this is harder for us to relate to. Okay, but this is optimal body temperature for a person. So you'll notice when it gets to a certain point, okay, and it gets too hot, it drops off very, very quickly. When it gets too cold, it slows down little by little by little. Okay, this steep drop right here, that is when the enzyme has denatured. It's no longer working. That's why it's just I'm done. Okay, enzymes, one of the things we need to be really careful when we're writing, when we are writing about temperature and enzymes, you cannot write that cold will denature your enzyme because it doesn't. As a general rule, the reason why the enzyme reaction is happening slower when it's cold is because the molecules are moving slower. And so the enzyme and the substrate just aren't coming together very often. And they eventually become frozen where they never come together. But that doesn't necessarily break the enzyme protein down. Where extreme heat or temperatures that are too high, at first they're great because as the molecules heat up, they move faster. And so as they move faster, they come in contact more often. And so you'd have more reactions happening. However, you'll reach a temperature where it breaks down the enzyme protein when it's too hot. And so it will denature. So when you're talking about temperature and enzymes, be sure that you address both ends and you address them differently. Cold doesn't denature your enzymes. It just slows the molecules down so the enzyme and the substrate don't come together as often. Whereas heat will speed it up to a point, and then it will, when it gets too high and completely out of that optimal range, it will denature the enzyme. And you can see how different organisms have different ranges. Like over here on the red, the optimal temperature of this heat-tolerant bacteria is, almost 40 degrees higher than ours. Okay? But that's what works for that particular organism. It's built for that. So pH is similar in the sense that the enzyme can denature. So we look at this graph over here on the right now. So that's referring to pH. And I know you guys haven't had tons of chemistry, so let's kind of review pH a little bit for a second here. Right? Remember, our pH scale goes from 0 to 14. Your graph here only shows it going to 10. Above 7 is basic or alkaline. Let's see if this will fit down here. Alkaline with an N there. And below 7 here is acidic. 7 is neutral. So right there, 7 is neutral. And so it's a little counterintuitive in the sense that, so for something to become a stronger base and more alkaline, it gets higher. And that makes sense because it's getting further away from 7. But things that stronger acids are actually lower because, again, I'm moving further away from 7. So the further I get from 7, the stronger my acid or base becomes. So an acid with a 0 or a 1 or a 2 pH is much stronger than an acid with a pH of 6. But a base with a pH of 13 is much stronger than a base with a pH of 8. So it's all centered around 7 and how far away they get from 7. Bases can be just as dangerous as acids. Bleach is a base, and bleach is very caustic. So when the enzyme gets out of its optimal pH, it will denature. That will definitely, those strong acids, those strong bases are definitely going to break that enzyme down. You can see this one on the left here, the black one, the optimal pH for pepsin. Pepsin is an enzyme that we find in our stomach. And because we have hydrochloric acid in our stomach, it makes sense that it works at this very low pH because it's in the presence of an acid. Trypsin, is not located in our stomach. It's located right as you come into the small intestine, and your small intestines are not built for acid. So your body actually secretes some bases right when that food comes out of the stomach to help neutralize those acids so they don't burn your intestines. And so the optimal pH for this one is a little bit above neutral, even though neutral is our normal pH of our body, because it's in an area where some bases are being secreted. So it depends on, again, where the enzyme is going to be as to what the optimal pH is. But whatever it is for that enzyme, if it gets out of that range, that enzyme can denature and then it won't work. Now, occasionally, the enzyme can denature and that can be reversible. Right? And that means exactly what it sounds like, right? That the enzyme could regain its function. It could regain its shape. So maybe you mixed in something that that helped the protein break apart, but it can retain its shape if those are removed. Okay? Think about hair. Okay? Think about the ways that we style hair. Hair is made out of proteins. Okay? And if I have curly hair or wavy hair, and I want my hair to be straight, I need to apply heat to it. And usually I need to apply a pretty hot heat to it. And when I apply that heat, that protein will denature. It'll break down and it will straighten out instead of being a curly protein. But then later, once I re-wet that hair, that protein will curl back up. That denaturation goes away. It's reversible in that case. Okay? So just kind of think about hair sometimes when you realize that this enzyme sometimes can be reversible, the denaturation of it. So with pH, though, the larger that difference is from the optimum pH, the more likely that enzyme is going to be to lose its function. And so the more likely it is to become denatured and be denatured permanently, the further it is away. So when we had that, if we have an optimal pH here, and let's say it's at 7, okay, we have an optimal pH here at 7. If all of a sudden my environment here is at a 10 and it's super basic, then it's going to be more likely for that enzyme to denature completely and not be able to be reversed back. Okay? What will happen is it will affect the hydrogen bonds. And so when those hydrogen bonds are affected, in the active site in particular, it makes the denaturation more likely to be permanent. So if your difference is small, let's say it was put in an environment of a 6, Okay? If your difference is small, those hydrogen bonds in the active site may not be affected, and therefore the enzyme could still possibly work for a little bit and then maybe even regain full function. So let's look at the concentration of substrate. This is your lab that you did with enzymes your freshman year, with the pressure and the tubes, and you soaked the disc in the catalase enzyme in the liver juice, and then you put the disc on the side of the test tube, and we measured the pressure of it breaking down hydrogen peroxide, and we changed the different concentrations of the substrate. Well, some of you did. Some of you changed the concentration of your hydrogen peroxide. All of you at one point changed the concentration of your enzyme. So we'll talk about that in a minute. But if you change the concentration of the substrate, that can affect the efficiency at the end of the enzyme. So if we look at this particular graph here, you can see here in the beginning is my initial velocity, which is another term for rate, right? And I could calculate the rate here by doing, I hope you're saying this at home, slope, right? And so what's happening here, okay, is my rate is going up really quickly, right? The amount of product that's being formed, okay, is shooting up pretty fast over a relatively short period of time. And so that's because my substrate concentration here, okay, I've got plenty of substrate. Time has just started. I've got plenty of substrate to turn into product. So my enzymes are just going. And then what will happen is you see my enzyme start to slow down. Right here where it starts to hit this curve, my rate starts to slow down. Right? My slope here is a lot smaller than it was back here. Right? It's not, my line isn't nearly as steep. So my amount of product that's being formed is slowing down. I'm not making as much product now in the same period of time, right? If I'm looking at this time window here, which is almost equivalent to this time window, and I'm not making nearly the amount of product that I was. And so my rate is starting to slow down here. And it's starting to slow down because as I'm forming more and more product, my concentration of substrate is going down. Right? I'm consuming the substrates. Enzymes aren't consumed, they're reused, but the substrate is being consumed. It's being broken down, and so it's not being reused. So as my concentration of substrate decreases, my enzyme, my speed that my enzyme is working at, my rate of my reaction, of my enzymatic reaction here, is going to start to slow down. And eventually it will become constant, where you have this flat slope here. Eventually that rate will become constant. It doesn't mean that the reaction isn't happening. It's just that the concentration of substrate has decreased. And so the enzyme can't be as fast, as it can't form as much product. Okay, you'll notice here my amount of product here that's being formed, I'm not really getting much of an increase in product here as time goes on because I have hit what is called my saturation point. And basically my enzyme eventually starts to run out of substrate to interact with. And so as that substrate concentration goes down, the enzymes start to run out of substrate to break down. So I'm not making any new product really here. Okay? And everything kind of plateaus and levels off. So changing my substrate concentration can affect that enzyme rate. And I can impact that a little bit if I increase the temperature, right? Again, if I increase the temperature, I get more interactions between the enzyme and the substrate. That's not going to completely overcome any kind of substrate concentration decrease. You know, eventually it doesn't matter how warm you make the reaction. You run the risk of denaturing your enzyme, and there just isn't any more substrate to collide with the enzyme. Okay? But again, like we said before, at first it's going to increase my rate a little bit, okay? And then it's going to drop very quickly, which is when that enzyme denatures. And you realize over here there's no quick drop over here in the cold because that enzyme did not denature. And the last thing we need to talk about is inhibitors. Again, hopefully you remember these from freshman year, competitive inhibitors versus non-competitive inhibitors. Okay, so inhibitors in general are going to prevent the enzyme from binding to its substrate. And they can do that one of two ways, in this competitive form or in this non-competitive form. If y'all remember the pool noodles that you worked with your ninth grade year, with the little C-shaped enzyme, and then we had the rings that fit in and out of it, and we put the little triangular shaped piece on one side of it to clamp it down. Okay, those were our inhibitors that were changing our active site. So a competitive inhibitor is going to look like or mimic the structure of the actual substrate. And so it is going to directly compete for the active site with the substrate. The competitive inhibitor has the same shape as the substrate, so it can bind to the active site just like the substrate would, and so it will fight the substrate for that active site. In your pictures here, the first one, you can see there's your normal binding. Here's your substrate. It fits nicely into that active site. That should work just fine. In the second one here, you've got this competitive inhibitor that was able to fit into the active site, and now this substrate no longer can fit into the active site. So it effectively is blocking the active site. They can't bind, and therefore your reactions will stop. If they bind to the active site with what we call a weak interaction, okay, then the binding could be reversible. But if they bind to the active site with those strong covalent bonds, like the kind of bonds that hold a water molecule together, then that usually makes the binding irreversible for a competitive inhibitor. The non-competitive inhibitors, they are not going to compete directly for the active site. What they will do is they will bind somewhere else on the enzyme to what we call an allosteric site. And so that allosteric site is somewhere other than the active site on the enzyme that this non-competitive inhibitor will bind to. However, when it binds to that site on the enzyme that's not the active site, okay, it will change the shape of the enzyme. If you remember with the pool noodles, we had the C-shaped enzyme. Right, remember we had this C-shaped enzyme and it had a little notch on the back side? Right, and we would put that little wedge in there. And when we put that wedge in there, it would make these close together. And then nothing could get into the active site. They were blocked from getting into the active site. So this little wedge over here wasn't binding directly to the active site, but it was binding to the allosteric site. Okay, which you can see right here on this purple enzyme changed the shape of the active site, and so the enzyme wouldn't be able to do that induced fit and wrap around that substrate, and so that would again in turn affect the enzyme's ability to catalyze the chemical reactions, and so it would decrease the efficiency of the enzyme. Okay, so both of your inhibitors are going to do the same thing. They'll decrease your enzyme efficiency. They'll just do it in two different ways. Okay, so we'll run some labs with enzymes and we'll look at some graphs and we'll work through this stuff in class as well.